Cells can switch between distinct modes of migration to move through different 3D environments. While still a postdoc in Ken Yamada's lab at the NIH, for example, Ryan Petrie discovered that, instead of forming flat lamellipodial protrusions, primary fibroblasts migrating through highly cross-linked extracellular matrices use Rho-A-dependent actomyosin contractility to generate blunt cylindrical protrusions called lobopodia.
And we found that the actomyosin contractility was actually being harnessed to pull the nucleus forward. And because of a diffusion barrier around the nucleus, what that forward movement actually did was to compress the forward cytoplasm and increase pressure specifically in the anterior cytoplasmic compartment. And it was this compartmentalized pressure that was driving lobopodial protrusions. So we named this the nuclear piston mechanism of 3D cell migration. And the question we had for the current manuscript was, are human tumor cells capable of using this mechanism that we discovered in human fibroblasts?
Petrie and colleagues placed HT1080 fibrosarcoma cells into the type of 3D matrix that induces the nuclear piston mode of migration in primary fibroblasts. The tumour cells, though, showed no increase in anterior intracellular pressure, indicating that the nuclear piston mechanism wasn't active. Like other tumour cell types, HT1080 cells use matrix metalloproteinases to clear a path through the extracellular matrix. Previous work has shown, however, that HT1080 cells can round up and switch to an amoeboid mode of migration when these extracellular proteases are inhibited.
So when we treated the HT1080 cells with the protease inhibitor, we saw the same switch in morphology where a proportion of cells switched from the elongated mesenchymal form to the rounded amoeboid form. But there was enough of both that we were able to measure pressure in each type of cell. And upon protease inhibition, there was no pressure change in the amoeboid tumor cells. And that was slightly surprising. But we then focused on the cells that remained elongated and polarized, the mesenchymal tumor cells, and found that upon protease inhibition in these cells, they began to generate the compartmentalized pressure, which was a signature of the nuclear piston mechanism.
To confirm that mesenchymal HT1080 cells used the nuclear piston mode of migration when protease activity was impaired, Petrie et al. Performed live cell imaging to compare the motions of the nucleus and the cell's trailing edge.
In the control cells, where pressure was low and uniform, the movement of the nucleus was in sync with the movement of the trailing edge, indicating to us, again, that the nuclear piston was normally off in these cells. However, upon protease inhibition, the movement of the nucleus and the trailing edge became uncorrelated, consistent with the fact that the nucleus was now being pulled forward by the actomyosin machinery to generate the large intracellular pressure to mediate lobopodial protrusions.
Furthermore, just like in primary fibroblasts, the nuclear piston mechanism in protease-inhibited mesenchymal HT1080 cells depended on actomyosin contractility, integrin-based cell adhesion, and on the nuclear envelope protein Nesprin-3, which connects the nuclear skeleton to vimentin intermediate filaments in the cytoplasm.
And we further extended our understanding of the molecular requirements for the nuclear piston mechanism by specifically knocking down not only vimentin intermediate filaments, but also lamin, a major component of the nucleus skeleton. And what we found was that both of these proteins were required for the cells to generate compartmentalized pressure in response to protease inhibition.
But why do the tumour cells switch to the nuclear piston mechanism upon protease inhibition? Moving the large, bulky nucleus through small pores in the extracellular matrix is the rate-limiting step in 3D migration. Matrix metalloproteinases usually help to widen these pores, and so, in the absence of protease activity, cells may switch to the nuclear piston mode in order to pull the nucleus forwards and force it through a tightly confined environment.
So we used two different types of type 1 collagen gels. We used rat tail collagen, which has a small pore size, and compared that to bovine dermal collagen, which has a larger pore size, where there was not a significant barrier to movement of the nucleus. When we added the protease inhibitor, we did see that in the small pore size rat tail collagen, not only did cells slow down significantly, but they also began to generate the compartmentalized pressure, indicating to us that the nuclear piston mechanism had become activated in that 3D environment. Doing the same experiment in the bovine dermal collagen with the much larger pore sizes, upon protease inhibition, cell velocity did not slow down at all, but we also saw that the pressure, in fact, did not change. So the activation of the nuclear piston in response to protease inhibition appears to occur specifically in environments where the nucleus is the rate-limiting step of 3D migration.
Petrie and colleagues wondered whether primary fibroblasts might forego the nuclear piston mechanism if their protease activity was elevated to the sort of level seen in tumor cells.
We were able to transfect primary human fibroblasts with the fluorescently tagged MT1-MMP, and we found that their ability to generate pressure in the 3D cell-derived matrix had been lost. We were able to rescue the pressure response by then inhibiting MMP activity with the drug GM6001. And so this experiment was crucial for us to really establish that MT1-MMP activity was controlling a switch between the low-pressure mesenchymal-type migration versus the high-pressure nuclear piston-driven 3D migration, and giving us good evidence, we think, that, in fact, protease-dependent mesenchymal migration and nuclear piston-driven 3D migration represent two distinct mechanisms to solve the same problem, and that is how to get the bulky nucleus through the confining 3D matrix. When protease activity is high, the cells are able to essentially chew their way through the material and enlarge the pores to make it easier for the nucleus to pass through. But when protease activity is reduced, the cells then rely on the power of myosin II to pull the nucleus through the unmodified 3D matrix, thereby generating the compartmentalized pressure that we now know as a characteristic of lamellipodia-based motility.
Petrie and colleagues now want to investigate how contractile actomyosin filaments are connected to the vimentin cytoskeleton and the nucleus in order to drive the nuclear piston and generate compartmentalized pressure. For now, though, you can learn more about how the nuclear piston mechanism of 3D migration can be activated in tumor cells in the paper by Petrie et al., published in the January 2, 2017 issue of the Journal of Cell Biology.